In the ongoing lecture series on plant tissue culture, in my previous lecture we discussed about immobilization of cells, wherein we discuss what is immobilization, why immobilization is done or why immobilization is an important aspect of plant biotechnology. We also discuss what should be the ideal character or desirable characters of an immobilization system, followed by different immobilization system depending upon the mechanism by which the cells are immobilized, and then we also discuss about certain other aspects of immobilization. Now friends, we must have discussed a term somaclonal variations over various lectures in the lecture series on plant tissue culture. Many a times I mentioned this term whenever we talk about subculturing or regeneration of plants under in vitro conditions or under tissue culture conditions. So friends, this is what I am going to discuss today about somaclonal variations. So what we are going to discuss in this lecture, that what are somaclonal variations. I will give you a very brief historical view of somaclonal variation, how these occur, who were the people who gave the term somaclonal variation. Then what are the causes of somaclonal variations in the plant system when they are regenerated through plant tissue culture. What are the mechanisms that are involved in occurrence of these somaclonal variations. Then how you can select the somaclonal variants once you have regenerated the plant under in vitro conditions, then how you can select whether a particular plant or the regenerated plant has somaclonal variation or not. Then how to detect these somaclonal variations in the plant systems, and in the end we will discuss about their applications. So coming on to what are somaclonal variations. Now as the name variation indicates, there might be certain variation in the regenerated plants, and these variations can be both genotypic or they can be phenotypic. So we can say that somaclonal variations are genetic or epigenetic changes that occur in plants regenerated from somatic cells under in vitro tissue culture conditions. Now when we say genetic and epigenetic changes, genetic is obvious because it will deal with the change in the number of chromosomes, or epigenetic will be dealing with the expression of those genes. Now these both genetic as well epigenetic changes, they are also related to certain phenotypic variations, and we know that phenotypic variations are nothing but the traits or the changes in the traits that can be observed. So we can say that these genetic or epigenetic changes also lead to certain phenotypic variations in the plants, and these phenotypic variations, as we discussed, are largely due to the chromosomal abnormalities or the molecular level mutations. So in the crux, I will say somaclonal variations are nothing but these are the variations of genetic or epigenetic or phenotypic levels that occur in the cells or somatic cells especially under in vitro conditions or the tissue culture conditions. Now these variations are also defined as genetic or epigenetic alterations, which are both qualitative as well as quantitative among the plants regenerated through in vitro tissue culture techniques. So when we say quantitative, then it means the number of chromosome may increase, the number of chromosome may decrease as well, and when we talk about qualitative, there might be certain aberrations in the chromosomes. When we talk about the phenotypic changes, there may be increase in the biomass, aerial biomass of the plant, there may be decrease in the pigmentation or hyperpigmentation in a particular plant, and so on and so forth. So these are some sort of changes that occur during these somaclonal variations. Now if we look into the historical aspects of somaclonal variation, it was first reported or observed by Braun in 1959, when he first observed the occurrence of tumor on the Nicotiana plants. And that tumor, when he further regenerated that particular plant using callus, and then using callus the plants were again regenerated, that tumor still occur in the regenerated generations as well. So he hypothesized that there are certain variations which remain stable in the upcoming generations also. So this was the first instance when the somaclonal variations were observed. Then Skirvin and Jenick in 1976, they worked on a species of Pelargonium, and they found that whenever the plants were generated from the callus, then there were certain variations in the regenerated plants, and therefore they gave that term as Kelly clones. In the same year, that is 1976, CB also worked on some of the phenotypic characters of the plant that were regenerated again from the calluses, and he found certain variations in the new generations. So he gave the term phenovariants. And in 1980, Shefford et al., they worked on the protoplast of a plant, and they found when the plants were regenerated from these protoplast, certain variations do occur in the regenerated plant, and therefore they gave the term protoclones. So by and large, if we see caliclones, phenovariants or protoclones, they largely emphasize on the variations, and the name varied with the explant material that was, or the regenerated plants from which they were generated, these names were given on those basis. It was Larkin and Scrocroft in 1981 who worked on Hordeum vulgare, and they describe certain genetic and epigenetic variations that were observed in the plants that were regenerated from the somatic tissues in tissue culture. And when they found these variations, they gave the term somaclonal variation. So these were the people who were responsible for discovering the term somaclonal variation. Now we move on to different causes of these variations. So as you can see that somaclonal variations can be, the causes can be broadly categorized into pre-existing variations which were already present in the explant material, and the variations or the causes that are due to the tissue culture conditions or in vitro conditions in which the plants are grown, under which the plants are grown. So first, talking about the pre-existing variations. These include chimeras. Now these chimeras are nothing but group of tissues which are having different genetic makeup. The cells are having different genetic makeup. Then second is chromosome aberrations and rearrangements. Again, if the explant material is having any kind of variation in the chromosome number or any kind of aberrations are there, in changes in the morphology is there, that will again lead to such kind of variations in the regenerated or the upcoming generation. Then cell cycle. We know that there are different stages of cell cycle, and again whenever the plant is grown through or the cell is growing through these different stages of cell cycle, there is certain variation in the either in the protein content, either in the DNA content, whenever these the different stages of cell cycle are there. So again these different stages also lead to variations. And the fourth is the transposable elements. Now these are nothing but these are the short DNA repeats which are mobile in nature, and therefore these are also called as jumping genes. And whenever these elements are activated in the plants, either due to any kind of stress or any kind of variation in the conditions, these also lead to such kind of somaclonal variations. Talking about the second category of the causes of somaclonal variations, that is those causes that are induced by tissue culture. First one is genotype of the plant material that is being grown under the in vitro conditions, type of the explant which we are using to develop the callus or the suspension culture, and from there you regenerate the plant. mode of regeneration, how you are regenerating the plant using either callus or suspension culture, that also leads to the generation of somaclonal variations. Then plant growth regulators, not only the type of growth regulator but also the concentration of plant growth regulator, the combination of different plant growth regulator will also lead to, may also lead to somaclonal variations. Number of subculturing, we have talked that prolonged subculturing leads to somaclonal variations. So number, how many subculturing do you perform when you are doing either callus culturing or suspension culturing will have an effect on the occurrence of these somaclonal variations. Then length of the culture period, how long you have maintained that culture. Obviously, when you are maintaining the culture for a longer duration, obviously you are going to subculture that particular callus or suspension, or you will replace the exhausted media with the fresh media over longer period of time, and eventually that will also lead to certain somaclonal variation. Why this happen, how this happens, we are going to discuss ahead. So coming on to the pre-existing variations, and first one as we discussed is the chimeras. Now, as I told you, these are the tissues having different genetic constitution, and these are developed from meristem with a layer of mutated tissue. So, when we talk about layer of mutated tissue, it means there might be a layer of cells which have a different genetic makeup in comparison to the cells of the other layers. Now, this mainly happens when we take one or more explant material which is having such kind of chimeric tissues. Now, this is obvious because if you are using explant which is already having cells of different genetic makeup, they will eventually lead to somaclonal variations in the upcoming generations. Now, I have taken a small example. It was found in a study that in banana, somaclonal variations were higher, around 5.3%, in the plants that were derived from the shoot tip culture, because in banana it has been found that shoot tip is rich in chimeric tissues, and therefore this was the major reason when the banana plant were regenerated using shoot tip culture, higher percentage of somaclonal variations were observed when these were compared with the plants that were derived from somatic embryogenesis. So, you can clearly see that the plants that were derived from somatic embryogenesis, the somaclonal variation was percentage of somaclonal variation was considerably lower, that is around 0.5% in comparison to 5.3% in the plants that were derived from the shoot tip culture. So, this is how these chimeric tissues lead to somaclonal variation and have an impact on the occurrence of these variations. Second pre-existing reason could be chromosome aberrations or change in number or their rearrangement. And if we look into the plant tissues, the most common type of aberration is the change in the chromosomal structure. Either the chromosome is fragmented or there is a reunion or exchange of the fragments of the chromosomes. Polyploid plants have also been observed wherein higher number of chromosomes is there, and such kind of plant materials or explant materials from such kind of plants is used in the tissue culture studies, they will exhibit greater chances of occurrence of somaclonal variations in those plants that are regenerated under tissue culture conditions. Third is the cell cycle. Now, we know that every plant has its own system of physiological system, own pattern of growth, and when we say own pattern of growth, that means the duration of the 4 stages of the cell cycle, they also varies in the species or at the cellular level, right? And therefore, irregularities or the errors during any of these phases have shown to cause somaclonal variations. Various studies have shown that any kind of error specifically during the M phase or in the replication stage, that is the S phase, induce chromosomal aberrations that eventually lead to the somaclonal variations. Fourth reason was the transposable elements, and as I told you that these are nothing but short mobile DNA sequences, which we also call them as jumping genes. They lead to certain kind of genetic mutations, which eventually leads to the rearrangement in the genes. Now it has been observed that when the plants are grown under in vitro conditions or the tissue culture conditions, such conditions they provide a favorable environment or conditions for activation of such kind of transposable elements or the jumping genes. And when there is an activation of such elements, there is a significant increase in the chances of developing somaclonal variations. Now we move on to the second category of the causes, that is tissue culture induced causes. So first one is the genotype. Now we know that every plant has its own genotype. Even different varieties of a same species will have its own genotype, and therefore it would not be wrong to say that some of the genotypes are more susceptible to produce these kind of variations in comparison to the other genotypes. So I have taken an example of Musa acuminata which shows higher variability than the plants which were derived from the Mucosa balbiciona. So here you can see that both the species belong to Musa genus, but when the plants are regenerated from different species, they show different tendency towards the variability or somaclonal variations. It's not only the amount of the variations, but frequency of regeneration also has an impact on the frequency of the occurrence of these somaclonal variations. So if we are continuously using a specific explant material for the regeneration of the newer plants, and that frequency of usage is pretty high, then we can say that the frequency or the chances of occurring somaclonal variation will be high. Second is the type of the explant material that is being used to regenerate plants under in vitro conditions. Now it has been observed that if you are using a highly differentiated tissue like roots are there, leaves or stems are there, they generally produce more variants than the explant which have less differentiated tissues like auxiliary buds or the shoot tips. Obviously there are certain exceptions in this case as well, because we discussed that shoot tip region has more chimeras in case of banana, which may eventually lead to the somaclonal variation. But by and large, if we look into the scenario, then the less differentiated tissue, if you are using as explant material, then the chances of somaclonal variations are less. But if you are using highly differentiated tissues for as explant material, then chances of somaclonal variations is high. The undifferentiated tissue such as pericycle or procambium or cambium as starting material or tissue culture reduces the chances of variation. So this is we have already discussed. Third factor was the mode of regeneration. Now the in vitro growth conditions can be extremely stressful for the plant cells, because we know that we are basically separating the explant material, extruding the explant material from a plant which is growing under normal condition, and then we are exposing that explant material to artificial conditions that are maintained during tissue culture. So there is a lot of physiological stress that occurs on those cells. Now such kind of stress leads to certain mutagenic processes in the cell, which eventually leads to the somaclonal variations in the upcoming generation. Now when we grow the plant under in vitro condition, lot of dedifferentiation and redifferentiation occur in the culture, because we take an explant material, we place it on a suitable media that will lead to the development of the callus, which is a dedifferentiated phase. Now this dedifferentiated phase is then again separated from that media and placed on another suitable media, where redifferentiation of those undifferentiated mass of cell occurs and organogenesis then occurs to regenerate the plant. So such kind of processes, regular processes, that are dedifferentiation, redifferentiation, taking the X plant material from that redifferentiated mass, again subjecting it to dedifferentiation. So such kind of changes, regular changes, will lead to both qualitative as well as quantitative changes in the plant genome, which will eventually cause to the somaclonal variations in the plant tissue culture. Then in vitro stress induce or activate transposable elements that also lead to somaclonal variations, because again these conditions put a lot of physiological stress onto the cells, which may activate those jumping genes in the plant, and we have already discussed that these jumping genes eventually lead to somaclonal variations. Now, if we look into the methodology of plant tissue culture, then we induce organ formation or organogenesis directly on the explant material or indirectly through the callus stage. And it has been seen that whether the plant has been regenerated or the organogenesis has been generated through direct or indirect method, both can lead to somaclonal variation. A study was conducted wherein Bacopa monnieri, that is Brahmi, was in which the organogenesis was initiated directly on the explant material, and in Tylophora indica, where organogenesis was initiated indirectly, that is through the callus, both the regenerated or the future generation of these plants exhibited some sort of somaclonal variation. So both direct and indirect organogenesis may always lead to somaclonal variations. Fourth is the plant growth regulators. We have discussed that it's not only the type of plant growth regulator, but the quantity or the concentration of a plant growth regulator or the combination of the plant growth regulator may lead to somaclonal variation. It has been seen that unbalanced auxin to cytokinin ratio can lead to polyploidy. We have already discussed that higher auxin concentration always cause increase in DNA methylation, which will again lead to the disruption of DNA and will eventually lead to mutations and somaclonal variation. So the proper concentration and combination of plant growth regulator should be there. Similarly, type of auxin also cause somaclonal variation. I have taken certain examples like somaclonal variations were higher in caladium culture on a media containing either 2,4,5 trichlorophenoxyacetic acid or 2,4-dichlorophenoxyacetic acid than when that culture was grown on 1-naphthalene acetic acid or indole 3-butyric acid. So these are nothing but different types of auxins that are utilized in the plant tissue culture. So here you can clearly see that it's not only the concentration of auxin or the combination of plant growth regulators, but the type of the growth regulator also has an impact on the occurrence of somaclonal variations. Fifth is the number of subculture. We have discussed number of times that rapid subculturing will lead to rapid multiplication of the cells and eventually high proliferation rate of the cells, which eventually leads to instability in the genetic system of those cells and increasing the chances of somaclonal variations. The duration of culturing or subculturing has a direct impact on the occurrence of somaclonal variation. Long period in culture increases the number of somaclonal variation in wheat. Similarly, increased subculturing of micro-propagated banana plants have also shown increased number of somaclonal variations. So friends, in the next lecture we will discuss about certain mechanisms and other aspects of somaclonal variations. Thank you.